Good morning. I thought you'd all be here on time because I even pulled the fire alarm so you'd get up bright and early. Anyway, I heard dopamine's your favorite transmitter, and hopefully I won't change that impression. But what I'm going to be talking about are different aspects of dopamine, especially as it relates to the physiology of the dopamine system and also how it impacts psychiatric disorders. So first I'll start talking about dopamine neurons and what makes them so unique, because they really do have a lot of unique aspects as far as what drives their activity and what they actually control. Then I'll give you a crash course on receptor pharmacology, just because this is something that you often aren't exposed to, but it's so critical for understanding drug action, especially acute and repeated drug action, and how it affects living systems. Then I'll go into dopamine neurons and their reactivity to stress, their involvement in depression, because everybody knows that serotonin and norepinephrine really don't have anything to do with depression. It's all dopamine. And finally, second part of the morning talk will be on dopamine and schizophrenia, which is what we've done most of our work on. Now, this is a tyrosine hydroxylase figure showing the dopaminergic neurons in the rat. This is the ventral tegmental area, and this is the substantia nigra. Now, first, the dopaminergic system is really compartmentalized. Unlike the noradrenergic system and the serotonergic system, where you have individual neurons projecting to widespread areas, which are thought to coordinate activity across regions. In the dopaminergic system, you have discrete projections, where neurons that project to the frontal cortex are different than the ones that project to the amygdala, or different than the ones that project to the nucleus accumbens, so that you actually get discrete control over function depending on these subregions. The substantia nigra, is the part that degenerates in Parkinson's disease. The original thought is that it controlled movement, but now we know it really is much more complex than that, and controlling sensory motor integration, and a number of other factors. As you move medially, you get into the ventral tegmental area. And this is actually a heterogeneous structure. The most medial parts of the ventral tegmental area project the ventral medial striatum, or a nucleus accumbens shell. And that tends to be involved in reward, drug abuse, affect, things like that. And then as you move more laterally, you start to get to areas that project to more associative striatum, that are involved in salience and integration of information. And we know that the dopaminergic system has a role across a number of disorders. Of course, in Parkinson's disease was where it was first discovered that dopaminergic systems were distinct from noradrenergic systems and have an important role. It has a long history in schizophrenia, especially the associative areas, in drug abuse, especially in the reward-related areas, and there's an emerging literature on depression in the dopaminergic system. So the dopamine system appears to have a major role in multiple brain functions and disease states. And of course, I really like the dopamine system. Now, I don't want you to think that I'm obsessed with dopamine. But studying dopamine does have its advantages. Now, this is actually one of the first studies that we did trying to identify dopaminergic neurons. This uses an old technique called glyoxylic acid histofluorescence technique. This is actually a modified to the original technique where you expose brain slices to hot formaldehyde vapor. And I hate to think the lab tragedy that led to this discovery. But when you expose catecholamines to these hot formaldehyde vapors, what you find are the monoamines tend to condense and form these fluorescent products. And some of the old studies by Dahlstrom and Fuchs actually did this and went through the brain and classified all of these different glowing areas depending on the color that they were glowing and their location. And this is why the ones that were glowing blue-green contain norepinephrine and dopamine, and those were labeled A1 through A12. And the ones that were glowing yellow were of course, the serotonin neurons, And those are the ones classified as areas B1 through B9. So if you hear about, A9, A10, B2 cell groups, that's where that classification came from before they could distinguish them. Now, in order to try to identify dopaminergic neurons, what I did is to do in vivo intracellular recording, and I injected this neuron with L-DOPA, which meant it increased the dopamine content, and since it was a dopamine neuron, it could protect it from degradation. And when we did the histofluorescence, it was glowing a much brighter color than the background dopaminergic neurons. So we knew that we were recording from dopamine neurons. Of course, then when we went to other techniques, we used tyrosine hydroxylase immunocytochemistry. In this case, the neuron was injected with lucifer yellow. And then when we did a tyrosine hydroxylase stain, you can see the same neuron now showing the combination of the lucifer yellow and the tyrosine hydroxylase stain, so that again we could identify it as a dopamine-containing neuron. Now, dopamine neurons have a lot of unusual properties. They have these very long dendrites that extend for hundreds and hundreds of microns from the soma. The axon that comes from the dopaminergic neuron actually doesn't come from the soma. It comes off of a major dendrite. So here's the major dendrite, and here's the axon that comes off of this major dendrite. Now, because this axon initial segment is a distance from the soma, it's not necessarily at the same membrane potential that the soma is at. And this contributes to some of the unique characteristics. Here's an intracellular recording from a dopaminergic neuron. So what you see is the neuron fires. There's an after hyperpolarization caused by calcium that comes in during the action potential. But after hyperpolarization ends, there's a slow depolarization mediated by a slow leak of sodium and calcium into the neuron that depolarizes the neuron and brings it up the spike threshold. Now, most thresholds, initial segment thresholds in the brain, are somewhere around minus 55, minus 60 millivolts. Here, it's between minus 30 and minus 40 millivolts. Now, why is this? Well, this is measured at the soma. The idea is at the axon initial segment, it is at minus 55, minus 60. But the slow depolarization depolarizes the soma before it gets to this point, so that this point is the axon initial segment really is its spike threshold, but when you measure it at the soma, it looks different. If you give something like 4-aminopyridine that makes the neuron more electrotonically compact, then this threshold drops down to where you would expect it to be in a normal neuron. Now, because of this, the action potential actually takes off from different membrane potentials at the soma, because in between the initial segment and the soma, there are a lot of GABAergic synapses on the dendrite, and they're going to change sort of the leakage between the initial segment initiation spike and the soma where you're measuring the current. So sometimes these spikes will take off at more depolarized potentials, in which case the whole neuron is more depolarized and the neuron becomes more broad. Sometimes it takes off at a more hyperpolarized potential, which makes the neuron more narrow. So when you're recording from a neuron, what you find are the action potentials in a single train vary in amplitude and duration, which is really unusual. And it's almost diagnostic for these neurons, especially in vivo where they're getting all these different inputs. So this is the intracellular recording of the action potential. This is the axon initial segment spike. Again, it's small because it's occurring at an electrotonic distance from the soma. This gives rise to the somatodendritic spike. These are masses of calcium spikes that are all being generated in the dendrites and lead to this broad action potential, which then leads to the after hyperpolarization. Now, to get from an intracellular action potential to an extracellular action potential, what you do is you differentiate this with respect to time, and that's what this differential is here. You do this because whenever you're looking at this intracellular action potential, you're looking at voltage. When you're outside of a cell, what you're looking at is the current generated by the action potential going across the resistance of the electrode. And when it goes across the resistance of the electrode, you're actually measuring the voltage drop due to the current. And of course, current is the first derivative of voltage, dV dt. And that's why the extracellular action potential is a differentiation of what's happening intracellularly. So the peak is the zero cross. The fast upward phase, this is the maximum rate of change, and this is the maximum rate of change of the downward phase. Now, if you use these criteria, it's actually very easy to identify the neurons. If somebody's recording from a dopaminergic neuron, as soon as I walk into the room, just from the sound coming out of the audio monitor, I can tell that it's a dopaminergic neuron. This is provided you don't overfilter them. If you notice, these are biphasic action potentials. There are no triphasic dopamine action potentials. If you see a triphasic action potential, you're overfiltering. When you overfilter, you differentiate it again and you get this flip. And what that does is it completely messes up your ability to look at what is the actual duration of the action potential. Now, how do we look at dopaminergic neuron population activity, the activity of neurons across, let's say, the ventral tegmental area? Well, if you record from a single neuron, you really don't get a complete picture of what's happening in the dopamine system. Because what we know from a number of studies, both in vivo anesthetized animals, as well as in freely moving animals, only about half the neurons are firing spontaneously. Half of them are non-firing. The ones that are firing at different firing rates and firing patterns. Now, each of these parameters are controlled by different afferent circuits and have different functional impacts. To try to understand this, what's going on, what we do is pass an electrode through either the substantia nigra or the ventral tegmental area in multiple tracks in a preset pattern, and basically count the number of neurons that we see firing in each electrode track. And that gives us a measure of what we call population activity, or the proportion of neurons that are active. When we find active neurons, we look at how fast they're firing, and also look at their firing pattern. And again, these all have different implications with respect to what's happening in the system. The first, whenever we look at firing pattern, in the baseline state, dopaminergic neurons fire in these slow, irregular pop-up-pop type of firing patterns. But studies by Wolfram Schultz and a number of others have shown that whenever an animal is exposed to a behaviorally salient stimulus, you get these bursts of action potentials. So these bursts of action potentials are really the rapid, phasic, salient output of the dopaminergic system, the thing that's probably most related to fast behavioral processes. Bursting alerts the brain that an important event has happened. Now, bursting is what we call phasic dopamine release. Whenever you get these bursts of action potentials, what you get is an enormous dopamine release intrasynaptically. It stimulates, especially in the ventral tegment, or in the ventral striatum, the dopamine D2 receptors that are mostly concentrated ventrally within the synapse. So burst firing promotes massive dopamine release in the synapse. And studies out of Mark Whiteman's group suggests that the concentration of dopamine within the synapse can reach hundreds of micromolar to millimolar concentrations, which means it completely saturates the D2 receptors with an action potential. But the thing is, because it's in high concentration and uptake processes are concentration dependent, you get a very rapid removal from this place within the synapse. So the burst firing event is a very rapid transient event. So this mode of release is dependent on phasic dopaminergic neuron burst firing. Burst firing seems to be driven by glutamate, especially from the area of the pedunculopontine tegmentum. This is a glutamatergic brainstem area that provides glutamate and acetylcholine to the dopaminergic neurons. If we activate this area by injecting the GABA antagonist bicuculin in vivo, what we see is a selective increase in burst firing without a change in number of neurons firing or in how fast they're firing on average. So this phasic bursting is driven by this glutamatergic input, but the glutamatergic input acts on NMDA receptors. Now, NMDA receptors have a unique property, in that the neuron has to be depolarized for the NMDA stimulation to have an effect. If the neuron's hyperpolarized, there's a magnesium block of the NMDA channel and nothing happens, which means firing neurons burst fire, but non-firing neurons don't change state. Now, this contrasts with what we call tonic dopamine release. Tonic is sort of the baseline activity of the system. This is driven by the slow single spike firing, driven by the pacemaker conductance that's broken up into an irregular pattern by the different GABAergic inputs. This causes dopamine release that's modulated presynaptically by glutamatergic and cholinergic inputs, but it's easily measured extrasynaptically. It probably occurs intrasynaptically, but in this case, the concentrations are very low, on the order of tens of nanomolar. So if you stick a dialysis probe into the striatum and you measure extracellular dopamine, you'll see it at between 5, 10, 15, 20 nanomolar. And that's completely dependent on the number of dopaminergic neurons firing. The more neurons firing, the higher the extracellular dopamine. So this mode of release is dependent on the number of dopaminergic neurons firing. Now, what regulates whether a neuron is active or not? Well, when we were doing in vivo intracellular recording from these neurons, we would often come across non-firing neurons. And whenever we penetrated them, you heard this constant rumbling in the background. And the rumbling is because these neurons are under a constant bombardment with GABA coming from the ventral pallidum. Ventral pallidum is a GABAergic area, fires in very high frequencies tonically, and holds these neurons in a non-firing state. In fact, if we inactivate the ventral pallidum, what we find is that the entire population of dopaminergic neurons now starts to fire. This seems to be controlled by a different system. So the ventral pallidum holds this system in an inactive state. But if we activate the ventral hippocampus, and here I'll talk about the ventral hippocampus or hippocampus subiculum in rat, which is analogous to the anterior hippocampus in humans. And this has to do with what happens during phylogeny. In the rat, the hippocampus has this dorsal ventral orientation. But as you go up the phylogenetic scale, the hippocampus slides into the temporal lobe. So what used to be the ventral limbic hippocampus is now the anterior limbic hippocampus in humans. If we activate this area, what we get is a selective increase in the number of neurons firing, more than a doubling, essentially causing all the neurons to fire, without a change in average firing rate or burst firing. We know that it's working through this pathway because if we block the hippocampus to accumbens glutamate pathway with kynurenic acid, we don't get this effect. And again, if we actually block the pathway here with bicuculin and saclofen for GABA A and GABA B conductances, we also prevent the effect.
Do they synchronize their firing rate when they spike all together, or do they kind of remove the inhibition?
They synchronize in clusters. So if you were going down with a multiple electrodes, you wouldn't find synchronous firing. If you go down with a single electrode, you'll often find neurons right next to each other that are actually firing synchronously. If you look at these areas, what you find is the dendrites tend to bundle, and the bundles are then surrounded by these glial sheaths. And we actually have evidence that there are gap junctions within these bundles of dendrites that we think actually coordinates firing. So let's say you have these individual clusters of neurons, and there's one neuron firing in each cluster asynchronously. But then the demand goes up, so you need to have more neurons fire. Well, in this cluster, you seem to activate neighboring neurons that activate through these gap junctions and probably have a slightly higher threshold. So now you start to recruit more neurons firing synchronously within like 5 to 10 neuron clusters.
So, and what is the role of D1 receptor in this circuit?
That gets more complicated.
D1 receptor can be.
So, I mean, the D1 receptor doesn't have a big impact within the VTA itself. There are D1 receptors on the GABAergic processes that are going into the VTA. But with respect to the postsynaptic site, you have a much different effect. So there's a difference between dorsal striatum and ventral striatum. In dorsal striatum, D1 receptors are more intrasynaptic, and D2 are more extrasynaptic. In ventral striatum, D2 are more intrasynaptic, and D1 are more extrasynaptic. In my longer talk that I cut out to make room for other stuff, what we find is that the D1 receptors seem to be better activated by the phasic bursts, and the D2 receptors tend to be better activated both by the phasic and the tonic systems.
So difference in distribution of D1 can determine the activity of the different parts of the system.
Yes. Yeah.
So gap junctions work through diffusion. How do you propose this would be like an active mechanism where you could control the firing?
How does...
So you said that you believed it was gap junctions.
Yes.
And how can that be like an active process where you can control except for diffusion?
So exactly what is controlling the gap junctions, whether they open or close, isn't clear. We know if you give a dopamine antagonist, you all of a sudden see a lot more neurons firing synchronously, but there are also a lot more neurons firing. Whether it's activating the gap junctions or whether it's actually something that's just increasing the number of neurons firing so that you're more likely to see the gap junction effect really isn't clear. So that's something we still need to investigate. Yeah.
Some neurons, when recorded in vivo, are extremely regular. Like you can set your clock to them. And what makes them so regular?
So they aren't usually regular unless you've manipulated the system. If you look at dopaminergic neurons in vitro, they're pacemakers. And the reason they're pacemakers is because you have that pacemaker conductance. Just like in the cardiac, the pacemaker conductance brings it up the threshold, it fires, you get a calcium-activated potassium conductance, hyperpolarizes it until you get the next action potential. And that's a very regular pop, pop, pop. In vivo, that's broken up by all the different GABAergic inputs coming from the ventral pallidum, coming locally, coming from GABAergic neurons that are projecting to other regions that have collaterals to the dopaminergic neurons, and that breaks it up into an irregular pop, 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 pop type of pattern. If you do a manipulation that decreases those inputs, striatal lesion, GABAergic blockade, even damage to the system, then you start to get the very regular firing. So what we have is this control in the number of neurons firing from ventral pallidal inhibition. But if we activate the hippocampus, it activates the ventral striatum, inhibits the ventral pallidum, and causes all of these neurons to start firing at their normal baseline firing rate and firing pattern. So what we have is a compartmentalization of the dopaminergic system. We have this massive dopamine release in the synapse mediated by burst firing. This is fast acting because the escape from the synapse is restricted by the dopamine transporter, both from the dopamine transporter on the terminal as well as by a neutral amino acid carrier that's a low affinity, high capacity carrier, which means when there's a lot of dopamine, it very rapidly removes it. When there's a little bit of dopamine, it's not so effective. This you really can't detect with something like microdialysis. If you put a microdialysis probe in and cause the neuron to burst fire, you don't see a big change unless you block reuptake, and then you see an enormous change. But you can detect it by raclopride displacement. And of course, this is what's used in PET imaging. Raclopride is a low-affinity dopamine antagonist that binds to D2 receptors, but not very strongly, and you can radio-label it. So you can give a human radio-labeled raclopride to bind to these receptors, and then phasically activate the dopaminergic system by giving some kind of a rewarding stimulus. You even see it with people playing video games. That causes a massive dopamine release, displaces the raclopride, and you look at the drop in radioactivity. And the drop in radioactivity is proportional to this intrasynaptic dopamine. And this is modulated again by this glutamatergic input, mostly coming from the brainstem. The tonic, it seems to be controlling a lot of the extracellular dopaminergic influence. So this influences large ensembles of striatal neurons through volume transfer. It's very low amplitude, 10 to 20 nanomolar. So it's not going to be affecting this intrasynaptic much at all. But what it will do is hit presynaptic terminals. It can hit dopamine presynaptic D2 receptors. It can hit D2 receptors on frontal cortical inputs. So it can very strongly modulate the frontal cortical input. It's longer lasting, since it's not as strongly affected by reuptake processes. And it is detectable with microdialysis. And in humans, it also seems to be a measure of fluorodopa. Now, fluorodopa is a measure of synthesis capacity. Fluorodopa is taken up into active dopaminergic terminals. Because it's taken up into active dopaminergic terminals, the more dopaminergic neurons firing, the more fluorodopa uptake there is. So it does seem to correlate better with the number of dopaminergic neurons firing than individual neurons showing fast phasic events. And this isn't as heavily influenced by reuptake. And again, this is modulated by the ventral hippocampal output. Now, this begs a question. If phasic dopamine releases dopamine into the synapse in millimolar concentrations, how can a systemic dopamine antagonist that reaches brain levels of hundreds of nanomolar to micromolar concentrations ever block the dopaminergic concentration in the synapse? Okay, this is me asking you a question.
You say that it's much more than the available receptors in the synapse?
There's way more dopamine than there are receptors available.
Okay, so then it's enough to block those to see the induction effect.
It all depends on receptor dynamics. And what Trevor twisted my arm into doing is going into my lecture mode to tell you something about receptors. Because the problem is, whenever you take these things into consideration, you have to remember that the brain is not a steady state system. If you were to mix millimolar concentrations of dopamine with micromolar concentrations of an antagonist and a beaker full of receptors, you would essentially get no blockade. But that isn't the way the brain works. So let's first talk about receptors. Let's say you have a membrane, you have a certain number of receptors. You want to look at binding of transmitter to receptors. So you add a radioactive ligand. And as you increase the concentration of the ligand, and you look at the amount bound, what you find is this. Now, why is this? Well, that's because it's binding to more than just receptors. Whenever you add this, it starts to bind just to the membrane sites outside of the receptor regions. This is called nonspecific binding. Now, to try to understand what specific and nonspecific binding is, you have to make some simplifying assumptions about receptors. And the simplifying assumptions is that the receptors are specific, and especially stereospecific. So they can only see one optical enantiomer of the molecule. They're also saturable, and they're reversible. Now, when you take this into consideration, they're saturable because there's only a certain number of receptors, and it's reversible because the ligand can bind to the receptors and then come back off the receptors. So what you do is you add your radio-labeled ligand, which is going to stick to receptors, and also to these nonspecific sites. You then come in and wash the membrane with a 100-fold excess of non-labeled ligand. What that's going to do is to displace all of the radio-labeled ligand from the receptors, but not from the nonspecific sites. Now when you do the same kind of thing, you get your classic binding curve, where this is percent bound, this is the log of the ligand that's added, here's your 100% bound, and 50% bound, which is defined as the KD. The KD being the dissociation rate constant. Now, the important thing to remember is that these parameters that I'm talking about are kinetic parameters. So let's take an example. You have a membrane, and I'm just going to use very simple numbers here. There are 10 receptors on the membrane. And let's say you've got 10 radio-labeled ligands. How long it takes a ligand to find a receptor is completely dependent only on the concentration of receptors and the concentration of ligands. It's like, blindfolded men and women mingling in a gym. The chances of a blindfolded man finding a blindfolded woman depends on the concentration. If there are only 2 of you in a field, it's not very likely. If there are thousands of you in this room, it's very likely, just by Brownian movement. So how long it takes it to find a receptor is going to depend a lot, again, on the concentration of receptors and on the concentration of the ligands. Now, once it finds the receptor, say you bump into the woman or the man, and you take off your blindfold, and you find that they don't speak the same language as you, or they're not very interesting, or you're not very attracted to them, not a very high affinity. So, you say, well, hi, I'm going to get some coffee. But on the other hand, if they're your perfect mate, you're going to sit there and stay around for a while. Well, this is the same kind of thing. When these ligands find the receptors, how long they stay on the receptors is a function of their affinity for the receptors, how well of a match there is between the ligand and the receptor. So the amount of time it takes to find a receptor, let's say it takes 10 seconds to find the receptor, just to give you round numbers. But because it doesn't bind very tightly, in 1 second, it comes off the receptor. At any given time, at steady state, what's the proportion of receptors that are going to be occupied? If it takes 10 times as long to find the receptor as it stays on a receptor.
About 1/10 the population.
About 1/10, right. Now let's say you have a higher affinity ligand. Same situation. 10 seconds to find the receptor, and it stays on the receptor for 10 seconds. At steady state, what proportion of the receptors are going to be occupied? About half, because it takes just as long to find a receptor as it does to come off the receptor. Now let's say you have the case where it still takes 10 seconds to find the receptor because it's the same concentrations, but it stays on the receptor for about 100 seconds. At steady state, what proportion of the receptors are going to be occupied? About 90%, right? Because it stays on the receptor for 10 times as long as it takes to come off the receptor. So let's go into some scary mathematics now. And don't worry, I hate math, so the time it takes to find a receptor, again, is purely dependent on the concentration of the receptors and the concentration of ligands. So here's your concentration of ligand plus your concentration of receptors, and this gives you your drug receptor complex. This is associated by a rate constant. And the rate constant is basically a fudge factor because you have moles, moles, and moles. And this gives you the number of moles per second. So the concentration of the ligand times the concentration of the receptor times this rate constant gives you the rate of association. If you increase the amount of ligand, the rate of association goes up. If you increase the amount of receptors, the rate of association goes up. If you increase both of them, the rate of association goes up because you're more likely for a receptor to find, or a ligand to find an unoccupied receptor. Now, once you get this drug receptor complex, then it has to go to free drug plus free receptor. And this occurs by a different rate constant, K2. In this case, drug receptor complex times K2 gives you the rate of dissociation. So this, K2, is the dissociation rate constant. The dissociation rate constant is dependent purely on the affinity of the drug for the receptor. The lower the affinity, the faster it's going to come off. The higher the affinity, the slower it's going to come off. Now, at steady state, the rate of association and the rate of dissociation are going to be equal at steady state. If it's a high affinity ligand, most of it is going to be over here. If it's a low affinity ligand, most of it's going to be over here. But the rates are going to be constant and the steady state levels are going to be constant. So then what you do is you just do a simplification. And what you get is KD. KD is the dissociation rate constant. And this is ligand, concentration of ligand times concentration of receptor over drug receptor complex equals dissociation rate constant over association rate constant equals the rate of dissociation. And that's that magical KD number. Now, this is all fine, but there's a problem. In that whenever you do experiments, you don't know what this value is. You know what this is because you can measure it. You know how much you added. So how do you get this? Well, if you keep adding radioactivity, at some point you get to saturation. At saturation, all the receptors are occupied. So you know the total number of receptors. So the total number of receptors is equal to the number of free receptors plus the number that are bound in drug-receptor complex, which means free receptors are total receptors minus bound receptors. You get this, you plug it in for free receptors, and you end up with the very familiar equation, XR, drug receptor complex, over total number of receptors is equal to ligand concentration over ligand plus the KD. This is percent bound. Now, when you look at your binding curve, as you increase the concentration of X, of your ligand, when the concentration of the ligand is equal to the KD, you have 1 over 1 plus 1, or 1 over 2. So you have 50% bound, and that occurs at the KD. As you increase the concentration of ligand, this approaches 1, which is your total number of receptors bound. Make sense? Okay, now I'm going to throw a wrench into this. Let's say you give an antagonist. If you give an antagonist, you shift this curve to the right, just like you would expect. Why is it shifting the curve to the right? Is it changing the affinity of the drug for the receptor? What's happening?
More ligand is now required to get the same 50% occupancy because some of the receptors are occupied by the antagonist.
Exactly. Because remember, KD is K2 over K1. K1 is dependent purely on the concentration of free receptors and the concentration of drug. What the antagonist is doing is decreasing the rate of association by blocking the free receptors, which shifts this to the right, not by changing the affinity, but by changing the ability of the ligand to find a receptor. Now let's take something really confusing. And this you will find misstated in pharmacology papers everywhere. Let's take a dose-response curve. Let's say this is the locomotor response to a dopamine agonist. Now you go in and make the system supersensitive. You give a dopamine antagonist over a period of several weeks. Then you withdraw the antagonist. You make the system supersensitive because now it takes lower doses of the agonist to get the same response. But if you take this, grind up the membrane, and look at percent bound, they completely overlap. Why? What happens with supersensitivity? Does the D2 receptor increase its affinity for the ligand? No. You get more receptors. Because you have more receptors, percent bound isn't going to change. But what is going to change is the relationship between occupancy and response. So what you have, let's say, I'll just give you some made-up numbers, that you have 2,000 receptors, and it takes 1,000 receptors to get a half maximal behavioral response. And this is your response, log of the drug. Half maximal receptor occupancy is half maximal behavioral response. Full occupancy is full behavioral response. Now let's say you've gone in and you've doubled the number of receptors. Now you have 4,000 receptors. Just like before, at the KD, you have half the receptors occupied. I'm terrible at drawing these things. The thing is, at half the receptors occupied, you've already got enough receptors stimulated to give a maximal behavioral response. Because you only need 2,000 receptors occupied in the normal case to get a maximal behavioral response. But because you have twice the number of receptors, at the same drug concentration, you have twice the number occupied. Which means now it's shifting the dose-response curve to the left, not because the affinity's changed, but because there are more receptors available. And that's why with supersensitivity, the KD doesn't change, the affinity doesn't change. What you have are spare receptors. You have more receptors than you need to get this effect. For the same reason, people talk about autoreceptors being more sensitive than postsynaptic receptors. They're both D2 receptors, and they both have the same affinity. What's the difference? Spare receptors. You have more receptors, which means at the same concentration of drug, you have a bigger response because you have more receptors occupied. Now let's go back to our question here. Why does micromolar concentrations of an antagonist block millimolar concentrations of dopamine? What happens whenever you have an antagonist acting with a pulsatile release of dopamine? Before that pulsatile release occurs, the antagonist has bound to the receptor. And it's going to stay on that receptor for a period of time proportional to the KD, or proportional to its affinity. It doesn't care how much dopamine is out there. It's going to stay on there until that period of time is over, and it can come off. Once it comes off, it will be completely outcompeted by the dopamine. But dopamine's occurring in a brief pulsatile fashion. The higher the affinity, the larger proportion of that synaptic event is going to be occupied by the antagonist. So it's not a steady state system. If it were a steady state system, you would never get blockade. But it's a dynamic system. And that's why you can get low concentrations of an antagonist blocking massive concentrations of the endogenously released transmitter. Mm-hmm.
Could you get blockage at steady state if the affinity of the antagonist was tens of or hundreds of magnitude higher than the affinity of dopamine to the receptor? In which case, even if he sees this difference is also hundreds of magnitudes, that is overcome by the increased affinity of the antagonist.
Oh yeah, but you'd be dead too. Okay, so that's a quick foray into receptor dynamics.
I have another question. So, do we know whether when the ligand binds to the receptor and a lot of these GPCRs get internalized after receptor ligand binding, does the receptor take the ligand with itself instead?
That's a more complicated question. What you find, especially with things like amphetamine, is you tend to get receptor internalization. And the receptor will internalize with the ligand, and it can make into some problems because now you've broken your cardinal rule because it's not reversible, because it's internalized. And it all depends on how fast it's metabolized.
Is there a way that a receptor can change its affinity?
Yes. GABA receptors are a perfect example. So a GABA A receptor has a certain affinity for GABA. But if you give a benzodiazepine binds to an allosteric site, and it changes the conformation of the receptor to make it a higher affinity for GABA, which is why benzodiazepines are such safe drugs. because it's not directly stimulating the receptor, it's just changing the affinity for GABA. So you could take massive amounts of benzodiazepines and not die. But then you could go and get a shot of alcohol on top of that. You get a direct-acting drug-receptor interaction massively potentiated by the benzodiazepine, and you stop breathing. So.
Would that ever happen, like spontaneously or in a disease, that a receptor would just change its conformation?
So spontaneously, the benzodiazepine ligand endogenously is thought to be beta-carboline. Beta-carboline is produced endogenously. And it's actually an inverse agonist for the benzodiazepine receptor, which means it decreases the affinity of GABA for the receptor. And that's why beta-carbolines tend to be anxiogenic, whereas GABA agonists tend to be anxiolytic. Okay? Okay, you can wake back up now. So, what I've been talking about is this tonic-phasic interaction. In the normal condition, we think tonic and phasic dopamine work together to modulate limbic system function. The idea is by setting the baseline tonic discharge of dopaminergic neurons, the hippocampus subiculum, through the accumbens ventral palatal pathway, controls which subsets of dopaminergic neurons can be phasically activated into burst firing. So if we look at the number of neurons firing, we activate the subiculum, we get a big increase in the number firing. If we activate the pedunculopontine, we don't get much of a change. If we look at burst firing, we activate the subiculum, we don't get much of a change. We activate the pedunculopontine, we get a big increase in burst firing. But if we look at a different parameter, the number of dopaminergic neurons that are showing high levels of burst firing, activating the ventral subiculum has a small effect, activating pedunculopontine has a small effect, but when you activate them together, you get this massive effect because there are more neurons that are available to go into a burst firing mode. So this controls number firing. This allows firing neurons to burst fire. Burst firing is the signal, the behaviorally relevant phasic output of the system. But the hippocampus through the ventral pallidum is the gain, or the level of amplification of the signal. And this, we think, has a lot to do with the functional effect on the system. So let's say you're in a benign context. You're at home in a safe neighborhood behind locked doors, or you're in a crowded room with no air conditioning after a long night of partying, listening to someone drone on about dopamine physiology. You don't expect anything exciting to happen. So the hippocampus has the dopamine system toned down, not a lot of neurons firing. Now you hear a noise from the back of the room. Well, the noise is going to activate this glutamatergic input, but because there aren't a lot of neurons firing, you're not going to get a big dopamine release. So you hear a noise, you look around and see somebody fell asleep and fell out of their chair, and you turn back around and keep playing with your iPhone. But let's say you're in an activating context. You're an animal out hunting, where every noise is something that you could eat or something that's going to eat you. Now you're in a very charged state. All of the neurons are active, or a lot of the neurons are active, because the subiculum is driving this context-dependent activation. Now, the same stimulus, which is going to cause the same glutamate release, is going to cause a much greater dopamine response. So you're going to be keyed up in order to respond to the stimulus. And what you have is a balance between this activation of the system and the response that depends on the contingencies of the context, that balances out between the signal and the amplification of the signal. Now, about 20 more minutes? Yeah.
So this means that when the dopamine neuron is already tonically active, it's more likely to give a phasic response?
Right.
And what is the reason for that? And what is the
Because the glutamate acting on NMDA receptors will only cause burst firing in the firing neuron. If it's not firing, it's hyperpolarized. Right. Yeah.
Toby, just before we get on, perhaps I can ask a couple of questions at this point. So,
Does this count into my total?
maybe you'll get onto this and you can say that I'll answer it after the break. So this is one part of the system. Is the ventral spiculum the only top-down structure governing the population of dopamine neurons available? Point one. Is the PPTG the only glutamate input? I mean, prefrontal inputs must indirectly affect this. And what happens with other sectors of the stratum? You know, if I'm talking about dorsal stratum, projections there, is there a parallel circuitry, or is it all triggered by this initial ventral spiculum to show process?
Now, the dorsal striatal system hasn't been studied as well, but we still have the same thing where only about half the neurons are active. And it's actually easier to measure in that system because the dopamine neurons are so compact. And there you have the parallel circuits where you have the subthalamic nucleus providing the glutamatergic input and the globus pallidus providing the GABAergic input. With respect to number firing and burst firing, the subiculum and the pedunculopontine are the most potent. But there are still others that can influence it. I mean, Gary's published some stuff looking at other pathways involving, which areas? Yeah. BNST that are involved in these kinds of regulations. There's glutamatergic inputs coming from the hypothalamus. There's glutamatergic inputs coming from the frontal cortex. But with respect to the most potent effects, these are the two that give the most potent impacts. And I'll go into this depending on how depressed I can get. So what about stress? Stress has complex actions on the dopaminergic system. And stress, anxiety can have very different consequences. First, stress is a part of our everyday lives. We need to have stress. It keeps us from avoiding reckless endangerment. It gives us motivation. It can really help us to avoid dangerous activities. But when stress is experienced in excess, either in terms of intensity and duration, it can have deleterious consequences. And depending on the individual and the conditions, stress can lead to major psychiatric disorders, such as drug abuse, post-traumatic stress disorder, depression, and suicide. And what we found is that stressors can have very different effects on the state of the dopaminergic system that correlates with activation or depression of responses. So let's say we look at a very simple thing, a single foot shock, a noxious stimulus. What we find is if we look across the dopaminergic population, not a big effect. But there is an effect if we look at subpopulations. If we look at the medial areas that are involved in reward, we get primarily inhibition. If we look at the lateral areas that are more involved in salience, we get primarily excitation. But these are very brief responses on the order of milliseconds. But if you do microdialysis, what you find is that stressors increase dopamine release in postsynaptic targets for long periods of time. When we record from population activity, we find very different effects. So in contrast to single stimuli, if we give foot shocks over a period of minutes and then start looking at dopaminergic neurons, what we find is something interesting. In the medial area, we tend to get a big increase in the number of dopaminergic neurons firing. Not so much in the lateral VTA. But this only works for the first 90 minutes after the stimulation, which is interesting because this correlates with what you see with microdialysis. With microdialysis, you see about an hour-long increase in extracellular dopamine. We see about an hour-long increase in the number of neurons firing, which is something measured with microdialysis. Now, what other things can stress cause? Well, one area that really is potent at being activated and modulating stressors is the ventral hippocampus subiculum. Foot shock activates the subiculum. Locus coeruleus stimulation that we know is involved in stress and orientation responses activates the subiculum. The amygdala, an area involved in anxiety and stress, when it's stimulated, can cause subiculum and hippocampal neuron firing. And we know that the ventral hippocampus has a unique effect on the dopaminergic system as far as number of neurons firing. In fact, if we go into our case where we give foot shocks and activate the number of neurons firing, if we inactivate the hippocampus, we bring it right back down to baseline. So this change in tonic responsivity of the system depends on the stress activation in the hippocampus. Mm-hmm. Not if you're giving individual pulses sort of repeatedly. They're all going to be showing their effects. They might start to show less inhibition, but what we see is this recruitment of neurons that tends to happen independent of what's happening with respect to the individual firing rate of the neurons. So if you record from a single neuron, you're going to get a very different picture. It's when you record from the population of neurons that you start to see this recruitment. Yeah?
If the magnitude of the foot shock were to be increased, would you see faster recruitment?
We haven't done that. I think that you would see a bigger amplitude, but I don't know whether the speed of change is going to be that different. So what we have with an activating stressor is activating the hippocampus, activating the ventral striatum, inhibiting the pallidum, and causing this big increase in the number of neurons firing and increased responsivity. And repeated stressors or repeated foot shocks increase the responsivity of the dopamine system. So this would lead to a hyper-responsive dopamine system that we think contributes to anxiety states. On the other hand, stressors that are maintained for extended periods of time put dopamine neurons in a distinctly different state that we think may be more related to something like depression. Now, major depressive disorder is often called the common cold of psychiatry because it's an illness that affects 15% of the population throughout their lives. At any one time, as much as 5% of people are depressed. And it's twice as common in women as it is in men, which means every woman, 1 out of 5 women will have a major depressive episode sometime during their lives. Now, historically, serotonin and, as we heard from Gary, norepinephrine has been linked to depression. But the link between serotonin and depression has been based a lot on the serendipitous finding that drugs that affect serotonin transporters are effective antidepressants. But there's a problem with these drugs. You have to give these drugs for weeks to get an antidepressant response. But you increase serotonin levels the very first time you give the drug. And acutely increasing serotonin doesn't relieve depression. In contrast, when you look at the symptom profile, it's likely that the dopaminergic system plays a role in depression. Certainly mood is involved with, serotonin has an involvement in mood. Norepinephrine has an involvement in attention. But when you talk about things like lack of motivation and anhedonia, or an inability to derive pleasure from the environment, this is the domain of the dopaminergic system. So the dopamine system should have a role in depression as well. So what we did is to look at the dopaminergic system using a chronic mild stress model of depression. Now, a chronic mild stress model is where you intervene with repeated, mild, unpredictable stressors. You basically irritate the hell out of the rats for about a month or a month and a half. So one day, the lights are left on for 24 hours. The next day, the bedding is wet. A couple days later, the cage is tilted. A day after that, they're put into a restraint, and you randomly vary these. And what you find after this is something that's consistent with a lot of things like depression. And just like Gary talked about, you have this forced swim test. The forced swim test is a very simple test. You throw a rat in a bucket of water and see how long they swim before they give up. Rats don't drown. If you have a flood, the rats are going to be the only thing that's going to be left living because they float. But what they'll do is when you put them in a bucket of water, they'll struggle and try to find an escape. But eventually they give up and they just float. And that's thought to be a measure of behavioral despair. And the more depressed a rat is, the faster they give up and the more time immobile that they have. And what we find is after chronic mild stress, we get a big decrease in the number of dopaminergic neurons firing without a change in firing rate or firing pattern. Yeah?
Could they not just be a bit more tired? Could they not just be a bit more tired? I know that's a bit of a silly question. I mean, the stresses that you're putting on them are probably disrupting their sleep or disrupting many other factors that perhaps are not. I mean, obviously that you see in depression as well, but do you see what I mean? It's quite a broad.
Yeah. Are they just tired?
I mean,
it's
doing things that are disrupting many.
So you'll get a lot of arguments about exactly what forced swim test does. And a lot of people will argue whether it really has any relevance at all. The thing is it's a really good correlate. And it correlates with antidepressant response and it correlates with induction of depression. What it exactly means is another matter.
Some people think that the inability is simply a species-specific coping response. It's a program. Mm-hmm. They struggle, they float. Mm-hmm. It's not despair per se. But I agree with you, that's predict what some antidepressants seem to do.
Yeah. Another version that I know about interpretation of this test, it seems that in some animal, treatment that improve learning make them stop even sooner, and they say maybe because they are smarter and they know that they cannot escape and they just give up.
Yeah. That's what I would do if I were a rat.
Yeah.
Depression behavior phenotypes like sucrose preference?
So we have done social interaction. We don't believe in sucrose preference. First, Sprague-Dawley rats really don't show sucrose preference. It's actually a rat strain specific response. And second, everyone who shows sucrose preference cheats. Not a single one of them has an x-axis that starts at 0. Because if you did, this would be the difference. If you go to a depressed patient and say, do you want this blank piece of paper or this $100 bill? They're not going to say, doesn't make any difference to me. But if you tell a depressed patient, somebody's giving out $100 bills, but you got to get out of bed, you got to get dressed, we got to drive over there and stand in a big queue, they're going to say, I don't want to bother. It's that motivational aspect that's impacted. And when you start to look at effort-based decision making, then you start to see an effect. So sucrose preference, I don't, it's one of those things that everybody has to do, but I don't really think it means anything.
What about other models, let's say, stress, such as pinch tail or food restrictions, like diets?
It has to be random. It has to be unpredictable and over a long period of time. If it's predictable, they accommodate. So if we did light on 24 hours, repeatedly, they'll accommodate to that. If we do cage tilting constantly, they accommodate to that. It's the unpredictable nature that seems to lead to the depressed condition. Exactly.
So if you do this with stress and annoy them constantly, how long do you still see this effect after you stop doing that?
We can see this for several weeks. Now, interestingly, female rats are much more susceptible to depression. In fact, when we first started doing this, when we compared control and female rats, the female rats were more immobile to begin with. And it's like, are female rats depressed to start out with? Well, when you do the forced swim test, it's a two-stage test. You expose them to the water environment, and then the next day you do the test. That single exposure was enough to make the female rats depressed, so that whenever we did the test, they showed more depression. And after chronic mild stress that increased the mobility in the male rats, there was even a bigger increase in female rats. Dopaminergic neurons at baseline between male and female are the same as far as cells per track. But after the first forced swim exposure, the female rats are already showing a decrease in the number of dopaminergic neurons firing, which after chronic mild stress, the difference is even more exacerbated. So we're getting a decrease in population activity that's even more pronounced, even with mild stressors in the female rats. Now, what pathways mediate this attenuation of dopaminergic neuron activity? Well, for this, we turn to the clinical literature. And what we find from the clinical literature is that there's one area of the brain that tends to be very strongly associated with depression. Question?
Does it matter what neuroscience cycle the female rats are at?
We've actually looked across cycles. We haven't controlled for it, but when we make sure that the control in the stressed rats are in the same stages, we don't see a difference. One area that's been consistently associated with depression by Helen Mayberg is the subgenual cingulate, area 25. This area is hyperactive in depression. It's activated by sad memories. It's activated by tryptophan depletion. And anything that's effective at treating depression normalizes activity in this area. SSRIs, SNRIs, transcranial magnetic stimulation, electroconvulsive therapy, vagal nerve stimulation, deep brain stimulation, even placebos when they're therapeutically effective, and they have a 50% placebo response, normalize activity in this area. Now, the subgenual cingulate, area 25, is functionally equivalent to the rat infralimbic prefrontal cortex. And in fact, if we go into the infralimbic prefrontal cortex and activate it with NMDA, we also get a decrease in the number of dopaminergic neurons firing. And this occurs exclusively in the medial VTA, the part of the VTA that projects to the reward-related striatum. If we activate the infralimbic prefrontal cortex, we get this decrease in number of dopaminergic neurons firing. Another area that's been involved in depression is the basolateral amygdala. And interestingly, if we block the basolateral amygdala, we bring dopaminergic neuron firing back to baseline, which means the infralimbic prefrontal cortex seems to be working through the amygdala. In fact, if we activate the basolateral amygdala directly, we also get this 50% decrease in population activity, without much of an effect if we inactivate this area. So this suggests that the amygdala is driving this decreased dopaminergic neuron activity. How is it doing this? Well, there are a lot of pathways between the amygdala and the VTA, but one stands out, the ventral pallidum. And in fact, if we take this pathway and block the glutamatergic input from the amygdala to the ventral pallidum with kynurenic acid, we block the effects of BLA activation on changing dopaminergic neuron activity. Is this involved in the chronic mild stress response? Well, what we find is that it is. If we inactivate the amygdala after chronic mild stress, we restore dopaminergic neuron firing. Now, can we bring this back to the infralimbic prefrontal cortex? So what we have is this complex pathway. We know that activation of this decreases population activity. That's blocked with this. What about the infralimbic prefrontal cortex? If we inactivate the infralimbic prefrontal cortex, we also restore dopaminergic neuron firing back to baseline.
What should be the, let's say, neurophysiological or neurobiological cascade from ventral follow BLA, interneuron, GABAergic interneurons, and integration with dopaminergic neurons.
No, it seems to be ventral pallidum directly to dopaminergic neurons. This is actually a very potent direct GABAergic pathway that doesn't involve the interneurons within the VTA. Susan Cisak actually sees beautiful histological evidence for this massive input that she hasn't gotten around to publishing, but that's one of those wonderful non-published things that I would love to see out. I mean, this is a complicated pathway, but stress is a complicated disease and probably involves multiple pathways as far as induction and multiple pathways as far as relieving these kinds of symptoms. And again, if we inactivate the infralimbic, we restore activity in the medial component. So in depression, the medial VTA is decreased because of this influence of the amygdala, which means stimuli that you would normally want to associate with reward are not going to cause the dopamine activation necessary to allow this association to occur. And this is why we think that you get this anhedonic effect with these chronic mild stresses mediated through this pathway. So the consequence is a failure to activate to normally rewarding stimuli. So what we have is this dopamine activating circuit and a dopamine inhibiting circuit that normally are in balance with each other. But after chronic mild stress, this inhibiting circuit predominates. Why? Well, we think this actually goes back to George Kube's opponent process model. He has this idea that whenever you activate the dopaminergic system, you start to get this opponent process that homeostatically works against the activation, but is longer in duration. And interestingly, 30 years ago, he said, we think it might be the amygdala that's involved. We see the same thing. This is the circuits that's activated by the stress that activates the dopaminergic system. But after a period of time, this starts to come in to counterbalance it, but this pathway drops out. And when this pathway drops out, it's only the inhibitory component that remains. And if I had another 15 minutes, I could tell you why we think this occurs. But it actually occurs because there's a disruption in the hippocampal to accumbens pathway that actually switches LTP to LTD. And interestingly, ketamine, a fast-acting antidepressant, completely normalizes this pathway. Yeah.
What are the depressive symptoms based on what would be more relevant to the baseline level of dopaminergic activity? Because, you know, I mean, obviously everybody responds to stress by decreasing their population activity, but, you know, if the baseline is already low, that you're more likely to experience depressive symptoms?
Exactly. The difference is resilience. Some people, when the stress is removed, they're depressed for a little while, but they come back. But some people don't. And what's the difference? Well, there's been a lot of work that actually came out of Ming-Hu Han's group that suggests one of the big difference is the responsivity of the excitatory pathway. Whenever you increase this pathway, the depressive symptoms come on stronger and for a longer period of time. Yeah.
How can you evaluate the severity of stress on this pathway? All of the stress with moderate or severe, how can it affect on this system?
Stress affects a lot of systems. It affects the noradrenergic system, it affects the serotonergic system. What I'm talking about is the dopaminergic component of the depressive response. There's also the component that's, the infralimbic prefrontal cortex does way more than just modulate the dopamine system. So what I'm saying is that we think that this is what's responsible for the dopaminergic mediated anhedonia effects on depression. But all the other things related to depression, all the other systems that are involved in depression are probably involving different transmitters and different pathways, all drawn because of the stress response and the lack of resilience, but affecting different circuits.
We're going to have one more question and then break, okay?
Okay.
So now if you were to subject rats to the forced swim test again, if you were to subject the depressed rats to the forced swim test again, would inactivating the BLA one shot would
Yep. And anything that reverses this reverses that.
It need not be that when they are being subjected to the stressor, that you need to keep the BLA inactivated so that it does not change the balance of the system. That in one shot, BLA inactivation is enough to restore the balance.
It'll bring it back until that wears off, because it's the infralimbic that's overdriving the amygdala. It's not the amygdala endogenous activity. And this, by the way, this pathway works with the chronic mild stress model of depression, and it also works with the learned helplessness model of depression. So it seems to work across different platforms.
So they'll take a break.